I would like you to give us a brief background around the research, what the interest is, and what were you hoping to achieve. Help us lay that background.
What we wanted to look at is the human genome effect of individuals that are infected with HIV. And within these individuals infected with HIV, there are certain groups of them that are able to control the virus naturally without having antiretroviral therapy. So the small group of individuals are sort of having a long-term protective effect because of their human genetics. So we wanted to identify which human genes, which mutations are present within these individuals, and if we can identify them using human genome sequencing. So we partnered with the South African Medical Research Council in South Africa and with MGI, and we decided to look at whole human genome sequencing. And what we found was that there are unique mutations that are found within these individuals, and these individuals show a protective effect. So we were hoping to try and see what is the difference between a individual that's protected from HIV progression, as well as an individual that progresses towards AIDS and eventually demises from the death from the disease if they don't take the antiretroviral therapy. So the idea behind this was that could we identify the mutations that are playing a role and potentially use this as a tool to mimic what's happening on those individuals that are able to control the virus. So now we have these protective mechanisms that are found in these individuals. Can we mimic it in some sort of a therapeutic intervention that we can design in the future to protect other individuals in the future?
Thank you so much. I think it used to be, I would say it used to be hypothesized probably 10 or 15 years ago, and probably 20 years ago, that we could actually have some people that have these innate protection against HIV on their own. And it's interesting that your team is now actively actually bringing this and working on this and letting us have an idea of what the science actually says. So what is the journey so far? What have you found, and what can you share so far regarding your findings?
Yeah, so I think the important thing is that you're right. There have been studies that have been looked at this in the past, where we've found human genes that are playing a role with HIV disease progression, and certain individuals are more protected from there. However, the studies in the past were conducted mainly in the West, and it wasn't directly related to an African population. And what we're doing is we're looking at an African population that has a specific genetic makeup that contributes towards protection. So the study is now relevant within the individuals that are directly affected by the disease. So sub-Saharan Africa has the highest rates of HIV around the world, and we're looking at HIV amongst this population and trying to find what's unique amongst their genetic makeup. And the idea is that we now will know what's unique in those individuals with regards to the genetics that's found within them, and how can that show a protection against disease progression, and can we use this as a potential therapy in the future once we've identified the specific mutations that are playing a role.
Yes, burning science question here, but I think somebody like you would like this burning question I want to ask. Are you doing whole genome sequencing in which you are just looking at the entire picture of that individual's genes, or you have specific genes that have already been suspected to be associated with this feature?
So we're looking at whole human genome sequencing. So we're sequencing the entire human genome of that individual. So we do have specific targets that we are interested in looking at, but we analyze the data after we've sequenced the entire human genome and identified new genes. Now, the reason why we want to look at human genome sequencing is because it hasn't been examined in an African population previously. So we don't know what genes or mutations might be playing a role in an African population that could be protection. Studies have shown in a Caucasian population, there are specific mutations that associate with risk as well as disease progression. This hasn't been studied to a large extent within an African population. That's why we decided to look at it at the whole human genome level.
Interesting. Did you share some findings at AIDS 2024 conference?
No, so right now we haven't published the paper yet. So we haven't identified, well, we don't want to ruin the paper. So we don't want to say which genes specifically are the ones that we've identified before the publication has come out yet. But what I can tell you is that there is some unique aspects that we've identified. So there are unique genes or mutations that are present within individuals of African descent that were not seen amongst the Caucasian populations. So Caucasian studies have shown that there are specific HLA genes that are associated with protection. So the HLA is your human leukocyte antigen genes that are shown to be protection against HIV disease progression, such as your elite controllers. We show that those protective effects are not found in Africa, and instead there are other genes and other alleles within the HLA that do play a role as well.
Interesting. Now, when we look at the bigger picture for HIV control, I think we are looking at the drugs have already gotten so advanced in minimizing side effects and in improving the quality of life. We are seeing advancement in chemoprophylaxis in terms of prevention tools. Condoms have come to be well established. We have PrEP that has also been extensively accepted. We have new set of PrEP that does not require those frequent injections like the ones that currently have. So we've seen a lot of advancement and growth in the space of HIV tools. Why do you think we are just catching up on this? And do you think it can be ready enough in the last mile race in ending HIV as a pandemic?
Yeah, so I think that with regards to the treatment that we have out there, the treatment is actually quite important to stopping the new spread of HIV, because we have a statement issued by WHO, the World Health Organization, which specifically states that U is equal to U, which means undetectable is equal to untransmissible. So if you don't detect the virus on the person using our traditional assays, it means that they cannot transmit the virus to their counterparts. So having this knowledge, it's important to get everybody onto antiretroviral treatment and getting them onto antiretroviral treatment so that they suppress the virus to an undetectable level. However, having said that, the level of drug resistance amongst antiretroviral therapy is starting to increase over time. So despite our best efforts to try and curb the HIV infections, the drug-resistant mutations that are present within the virus is also causing a problem, because the person is not able to suppress the virus anymore because of the mutation, or the drug not working on that individual because of the mutation. So alternative forms of therapy are starting to become very important now. So different types of treatment methods, different types of treatment techniques are quite interesting to actually start looking at. So antiretroviral therapy specifically focuses on the actual virus themselves. But our research is now looking at the human genes and the human genetics and see if we can try and identify what's unique amongst people that naturally control the virus. Why are they able to control the virus naturally? And what can we learn from them in terms of mechanisms of protection that we can use to design better therapy in the future? So this type of research will allow us to do new types of therapy in the future, such as gene editing, also personalized precision medicine that will specifically look at certain individuals based on their genetic makeup.
Let's talk about this particular phase. I'm talking about this in terms of availability of technology to be able to pull what you're trying to do off. So I'd like you to connect something together. How do you identify people to screen for this? How do we know that within a large population, how do we, since we don't have a target that we are looking for, how do you know the members of the population to screen for this? And which progression in technology has made what you are trying to achieve possible at the moment?
So I think it's important to first understand the difficulty of getting elite controllers. Now, these individuals that have the ability to control HIV, the rules and guidelines of HIV has changed over the past several years. Because previously, if a person was infected with HIV, the CD4 counts would have to drop below a certain limit before a person was administered antiretroviral therapy. Now the guidelines have changed and they've stated as soon as a person's infected or finding out that they're infected with HIV, then that individual would have to start antiretroviral therapy immediately. So that person would then be able to suppress the viral load in that person because of the antiretroviral therapy that they're taking. Now, elite controller individuals are able to suppress the virus without taking antiretroviral therapy, simply because if they are on antiretroviral therapy, they could be suppressing the virus because of the drug or because of their natural ability to do that. So studying elite controllers is extremely difficult simply because we don't have that pool of patients that we can try and look at, simply because of the guidelines that allow individuals to get therapy as soon as possible. However, saying that, in South Africa, we have a population of 7.8 million people that are infected with HIV. And certain individuals do not take the antiretroviral therapy, but report being suppressed within this environment. So those individuals are the ones that we could study with regards to elite control. Of course, these individuals are counseled to telling them that to take the antiretroviral therapy, but they default with their therapy as well. And that allows us to study these individuals. So this group of individuals are quite unique to study. They are less than 1% in the population that are infected with HIV. So studying them is actually very, very difficult and very, very unique. So also the ability of finding elite controllers in the future becomes difficult because if individuals are taking antiretroviral therapy and more people are being encouraged to take their therapy, then identifying the controllers are going to be a difficult task.
Yes, I think I totally agree with you regarding the impact around signing people up for treatment and how that could actually have on the ability to determine who is actually an elite controller. There is also, I'm curious to know, there is this localization issue, which I think you mentioned, that there is still some differences between what you are seeing in South Africa and what has been reported elsewhere. If this uniqueness and wide gap becomes really, really prominent, then what is the reality of developing a potential product from this intervention? If it's so restricted to region by region, country by country, what is the reality of being able to actually develop a product that will serve that will help as many people as possible, since we are seeing different genes or different haplotypes or different manifestations? So what's the reality of going to market with that if it's so different?
Yeah, yeah, I think it also highlights the importance of looking at this, because it means that, the approach of one size fits all doesn't really work. The one pill approach, where every single person takes one pill to kind of solve the problem. Even if we come up with a vaccine or come up with a cure for HIV, and we decide to administer it as one pill for everybody, that's probably not going to work, because the differences that exist across populations. And what we need to do is we need to study the different populations specifically and see how that treatment is going to work. So we need to look across Africa. So Africa has the largest genetic diversity around the world. And we need to show if we've identified some specific protective effects, does that protective effects contribute the same across every single person in Africa? And if it doesn't, what are the mutations that are linked to that protective effects in each of the different populations? I think the first step is trying to identify that across the populations, and then developing a vaccine or drug or a type of therapy that will be able to be administered to different populations based on their genetic output.
Do you have what does the timeline for the HIV host genome project look like? Veron?
So we are in the final stages right now with the HIV host genome project. We're basically analyzing all the data. And I would say within the next few months, we should have a fairly concrete project that summarizes what the specific mutations are and how they associate with specific elite control status as well as rapid progressing status. The idea is that this would serve as a stepping stone or platform for other larger studies in the African continent. The idea is that we should look at genetics that are playing a role in HIV disease. And if we've identified specific mutations that we think are playing a role, does that translate across other African countries as well? And I think that's the important stepping stone that will need to happen. But what the study has also done is that it's also sort of laid the foundation for new type of research. And Rizwana can discuss in more detail about the 110,000 genome project, where we want to look across different populations and in South Africa and identify what the human genomes that associate with specific diseases. And since we've laid the foundation with the elite control study or the HIV host genetic study, the next stage is basically duplicating this across other diseases in South Africa that are of relevance.
I think you are widening it, you are broadening it. So I'm curious to know how much the proportion of these elite controllers in our population, how significant are they in the population that you've studied, and are there some unique geographical or cultural, or what are the common threads that you think you can share with me of relevance that uniquely defines these HIV elite controllers?
Yeah, so the elite controller individuals don't really have any specific type of ethnic background. They don't really have any specific characteristics that you can show The only aspect is that they are able to control the virus for extended periods of time uh, in uh, in in that setting. And I think that the main aspect about that is that elite controllers exist in basically a range of different type of settings. So they can exist in Caucasian environment, they can exist in an African environment and an Asian environment. Elite controllers exist basically everywhere in the world, but they found in higher numbers in South Africa simply because we have higher number of HIV infected individuals. So there was a partnership that was established by the South African Medical Research Council and MGI. And MGI partnered with the entity to help with the whole human genome sequencing. So the sequences that we are able to use, which was provided by MGI, allowed us to conduct the research in this environment. This partnership has grown and included University of KwaZulu-Natal as part of this partnership. So we have this trifactor partnership between SAMRC, MGI, and University of KwaZulu-Natal conducting this research where we look at whole human genome sequencing on our elite controllers, and that associates with protection amongst certain human genetics that are associated with elite control.
Okay, talk me through what the best case scenario would look like for this elite controllers initiative, and what is your broad vision and wish regarding this research and endeavor?
Yeah, so the best case scenario would be that we identify specific human genes that associate with elite control status. Once we've identified these specific genes, the next stage is to develop a specific therapy that might be able to control that gene. So let's just say that gene is associated with protection towards HIV. Now, in people that are rapidly progressing with the virus, do those individuals have less copies of of that gene? And can we increase the number of copies of that gene so that they get more protection against HIV? So we can basically change the person's genetic structure so that they be able to get protected from HIV simply by giving them better human genes to fight off the virus naturally. So that could potentially serve as a novel directed therapy according to the individual's genetics.
Let's say we are not able to achieve all of these wishes. What do you think this research and this endeavor can realistically add to science?
Well, so far we've already shown that the mutations that are present within African descent individuals are very different from individuals from Caucasian descent. So what we've identified so far is that the genetics are quite different in that extent, and that those mutations will or could play a role in disease control. So realistically, what we've identified is differences that exist across populations, and those differences associate with HIV disease in a specific manner, and that specific manner is unique to our African population itself.
So what's next for you?
So what's next is that I think we have to identify the mechanisms specifically to confirm that once the mutations that we've identified are associating with HIV disease, how do those genes or mutations actually work in controlling against HIV? And can we mimic that in a setting such that we can form a new therapy for individuals?
Then I'm curious to know, connecting genes with prevention. How do you think that is going to be proven? For instance, so how do you want to associate that looking at the current landscape around HIV pathogenesis, where people live their lives at risk. So how do you think that association can be established?
Yeah, so I think what we're looking at is more progression of the virus after you're infected with the virus. Prevention is a different aspect. Preventing against HIV with regards to human genetics hasn't been very well studied yet. So we're not sure how that actual mechanism will work in that setting. But looking at prevention against progression of HIV disease, that's something that we're looking at amongst HIV infected individuals. So trying to find out the specific genes and genetics that play a role with the progression of the disease is something that we've basically on the journey right now to identify. However, prevention of infection is something that's not really studied well, and that will require much more in-depth discovery to identify.
Probably still on this, how realistically this is going to work. I'm also curious to know how you can deal with confounding factors, non-genetic confounding factors. For instance, we can have a situation, we know identical twin studies that the environmental factors were what defined the differences in in different manifestations, and the arguments could be like, okay, this person that we say is an elite controller, yes, the gene is there, but the environmental factors, the lifestyle choices, several factors that are non-genetic will still be involved in whether this person eventually, like lifestyle or alcoholism or all those other lifestyle choices, which have been found to actually play a crucial role in immunocompromise or immunocompetent status of an individual, would actually be the defining factor and not just genetics. So how would you respond to that? And I know you're already thinking around it.
Yeah, so I think that it's important to understand that human genetics is one of three components that are responsible for a person's disease state. So the environment is, one of the components that contributes, like what type of healthy lifestyle the person lives, what type of food they eat, all of that type of information will contribute. What type of comorbidities they have. Do they have diabetes? Do they have tuberculosis? Those are all other environmental issues that will contribute towards the person disease itself, HIV disease. But in addition to that, the virus itself may also play a role. So HIV in that individual, let's just say we're looking at a person that is an elite controller. So that individual may be able to control the virus simply because they have a defective HIV that's infected them. So because they have a defective HIV, it doesn't replicate as well or doesn't replicate at all. So that's why the person's able to suppress the virus naturally. So that's, looking at the virus itself. And then the final component of the three different avenues is the human genetics. So the human genetics are the ones that could also play a role with regards to the protection of the disease itself. So these three different components together play a role in determining whether a person is progressing to HIV or not, depending on how they contribute to each of the different categories. In certain individuals, the genetics plays a larger role compared to the virus, because the virus may not be defective. The virus may actually be fine and replicating quite easily, but the person still is able to protect against HIV. The person may be having a completely bad lifestyle or environment, so they have also comorbidities as well. But the person's genetics may also protect them from the disease as well. So it's a combination of these three different factors that have to be taken into consideration when studying the effect. what we've done in our study is that we've looked at the virus itself, and we've looked at whether the virus is defective in those individuals. And we showed that in the elite controller individuals, the virus is not defective. The virus is still functional in those individuals. However, they are maintaining at a very low level of HIV.
Thank you for that clarification. You said you are using whole genome sequence, and since you are looking at members of the population at risk groups, what is the reality of being able to do whole genome sequence for loads of people without, what this requires if you want to go to the lab who do whole genome sequencing? So how are you able to, what has been simplified to make it easy to be able to do this at the front line, to recruit and of course to carry out this highly expensive and tech-laden procedure at that scale?
Yeah, so I think that one of the main things is that whole human genome sequencing is currently being done on certain projects in South Africa in a very, very limited capacity, simply because there's not many instruments that exist within South Africa. The 110,000 Genome Project is hoping to increase the genetic pool of knowledge within this environment and try and identify what are some of the genetic effects with certain diseases. But to answer your question with regards to seeing if this is feasible in the long term with regards to a patient coming in, getting a whole genome sequencing done and getting the genome analyzed and interpreted, I think we still have a bit of time away before that can happen, because the facilities to actually do whole human genome sequencing and offer this as a service to individuals that come in for testing will require a lot more capacity than what we have right now. A lot of the whole human genome sequencing that exists in this country is specifically for the purpose of research projects. The purpose of diagnostics is what you're referring to, where a person can come in and get the human genome sequenced and identify specific polymorphisms that might be playing a role with a certain disease. Does not really exist within the country right now. A lot of those type of tests would have been sent out of the country. So we won't be ready to do it, at least in the near future, with regards to this whole genome sequencing at the patient level, to report back to the patient.
What are your last remarks what are you looking forward to? Any last thoughts for me?
Yeah, I think that I'm grateful to the partnerships that we've created throughout this journey. And I think that, partnering with SAMRC as well as MGI has helped us achieve these goals of doing whole human genome sequencing in South Africa amongst this HIV infected population. And I think it sets the platform for new whole human genome research on other diseases. And I think that's where we should be looking at in the future regarding new types of studies, focusing on this technology that MGI has brought to the continent.
So thanks so much and enjoy the rest of your day.
All right. Thank you so much, Paul.